So, the first mock lecture is from the College of Global Liberal Arts, a lecture from Professor Powell. I will introduce him briefly. So, Professor Powell is an associate professor in the College of Global Liberal Arts at Ritsumeikan University. He received a Doctor of Engineering degree at Nagoya University. Currently, he teaches Introduction to Science and Technology, Introduction to Scientific Thinking, Ocean and Society, the Blue Bridge in Asia Pacific, and a special lecture on Sustainable Development Goals. His specializations include science and technology, renewable energy, biomass and energy, energy and sustainable development. Okay, so Professor Powell, I know I have been looking forward to hearing your lecture, so I will let you take things over from here.
Thank you. So let me share first my uh, PowerPoint. So let's start. Good afternoon, everyone. So our topic for today is are genetically modified foods, GM foods, good or bad? So you can call me Powell. I'm from College of Global Liberal Arts from Ritsumeikan University. And let me start the session. So our topic for today we will be answering the questions: what are genetically modified organisms? Why do we need genetically modified food? How do we engineer GM food, and are GM foods safe or bad? We will decide today. So let's start with what are genetically modified organisms? So genetically here refers to the genes which are made up of DNA, and a set of DNA, you call it a gene, and it has specific information. Now, modified there implies changes to the genes which are done in a laboratory using technology that we have, and I'll be discussing later on biotechnology. And then organisms refers to life forms. It could be plants, animals, bacteria, all in the animal or all in the life kingdom. Okay, so these are basically genetically modified organisms. You modify the genes of the organisms, and this video here, a short GIF, is showing the gene. Okay, now World Health Organization defines genetically modified organisms are as organisms in which the genetic material, the DNA, has been altered in a way that does not occur naturally by mating and or natural recombination. And the food produced from GMO are called GM foods. It could be GM plants or GM animals. When it turns into food, they are GM foods. Now, why do we need GM foods in the first place? We can definitely produce the traditional food, but why the need for GM foods? First, our growing world population. Now we have 8 billion people, around 8 billion people, and we are increasing, predicted to increase until 2100 before going down. But then still, we have absolute value of population growing. Okay, and there's a lot of people that we need to feed, and we need more food to feed more people. Aside from having more absolute number of people in the world, the growing number, we also aim to make everyone to have better lifestyle, so to have better income. And it entails that if we have higher income, it means that we also consume more food. So aside from absolute value of the growing population, just becoming richer, the population will increase its consumption of food without changing the number of people. So let's put pressure on the food supply. Also, we have we currently there's 2.3 billion people that are already moderately or severely suffering in food insecurity, and this uh, worsen during the pandemic. At least I think it's becoming this becoming is recovering a little bit, but GM crops have better harvest yield per area, and this can, okay, without increasing the expansion the farmland, just by using GM crops we can have better harvest yield per area. And also undernutrition. So let me share this video because there's a lot of still the population that suffers death due to undernutrition. So 45% of death among children under 5 years of age are linked to undernutrition. And GM crops can be designed to have more nutritional value compared to the traditional crops that we have. So let me share this UNICEF video from YouTube.
Bishibo alelo mpenjo kachakuma tukwa nayo jumwanza mumemi mwana mulala bishi mari uju jani uju jilani ju tabalo ko kwani janu mteke tanu mula nenda
do pinje suka mutanga men ba ke ko ni bintu bya kujya ife mu di mbande ni bintu bya kujya ka bien ngo
tous les enfants présentant la malnutrition, présentant des complications médicales sont pris ici gratuitement et on arrive souvent à les guérir.
So with the advancement of our technology today, this should not be happening, or at least already minimized to a level. But still, this is a common occurrence in many of the places in African continent. Okay, now aside from that, there's a decreasing farmable land already. So we have, okay, we are converting this farmable land to other uses. So from 1950, the population increases up to now, and then from 1950, the arable land decreases up to now. So it means that since our population is growing, we are using it for our needs. Okay, aside from converting it from farm to cities, for example. And if we use GM crops, we don't need to increase the farmland just to increase the harvest yield, because GM crops can have better harvest yield per area. Also, the impact of climate change is really becoming apparent recently. So there's, aside from accelerated urbanization, wherein the farmland now are malls, land desertification, salinization, and degradation, even land conversion, climate change, drought, and flooding of farmland, and of course, water resource limitation. These are the things exacerbated because of climate change and human activities that negatively impacts the supply of food. And we can always use traditional breeding, but then there's a lot of limitation of traditional breeding that we cannot compromise now, because developing new seeds adapted to the changing environment takes a very long time. When we use traditional breeding, it takes 10 to 15 years just for wheat. And We need to identify crops with the desired traits. It takes time, needs to find out if crops with desired traits are even sexually compatible. But if we use GM crops, it can be rapidly and easily designed to thrive in a fast-changing environment. And even what kind of nutritional value or enhancement do we need? So GM crops are really flexible compared to the traditional breeding method. So genetically modified crops can be developed to grow in various types of land, such as arid land, land with salty water, flooded land, and others that are now a reality because of climate change. New varieties can be developed rapidly and accurately and have higher harvest yield per land area. And also, we can design it to be nutritionally enriched, depending on what nutrition are we targeting. Now, how do we engineer GM food? Just to give a quick background about modifying plants through selective breeding, because we have been modifying plants, organisms since ancient civilization. So the farmers, so if you can see, through selective breeding, they are modifying these different plants. For example, this is the carrots here. This is the first corn, and this is the first tomato. Now, because of the selective breeding of bigger and bigger, for example, in the corn, more and more kernels, and then they are selecting the specific plants with more kernels and making them, then they create a bigger and bigger corn. So humans have been manipulating DNA in plants and animals already for millennia. So this is not a new process. These are also an example of how we modify plants. Okay, so this is these are kale, cabbage, broccoli, cauliflower, kohlrabi, Brussels. These are the different parts of this cabbage plants that we have been modifying. Now, how do we genetically engineer this kind of food? What's the difference between traditional breeding and genetic engineering? So there's three genetic engineering techniques that we use. So if you can notice, the genetic engineering here is what I am using, because we are genetically modifying plants already through selective breeding, but using biotechnology, we are genetically engineering. Okay, so that's why we introduce genetic modification as a term, but then in reality, it is genetic engineering, because genetic modification can be done naturally, okay, through selective breeding. So now I am using genetic engineering here. So genetic engineering techniques, okay, these are the biotechnology techniques. So there are three techniques: direct transfer of DNA using a gene gun, transfer of DNA using bacteria, direct editing of genomic DNA through CRISPR-Cas9. This is a very new technology, just this decade, 2005. So there's three main techniques. We will be briefly discussing this just to give you a quick background on this. Now, for the direct transfer of DNA, this is the gene gun here. So basically, it is literally a gun that has metal particles coated with DNA, your desired genes, included in the metal particles. That's the bullet, and then they are trying to shoot it on this cell, plant tissues, and hopefully, okay. So there's a luck here. If you're lucky, then you can successfully, okay, shoot the DNA of your target tissue, and then hopefully the DNA can incorporate, okay, in the target tissue. The same thing as for this transfer of DNA using bacteria, you will have plasmid here inputted into the, or input in the agrobacterium, and then later on you will allow this agrobacterium to infect this tissue, and hopefully that agrobacterium, when infecting the tissue, can transfer the DNA that you want. So there's still a luck there, okay? Hopefully you're lucky, and then you grow, and then you check if if your target gene is expressed in your DNA, in your plant, and then of course, you have to grow it to know if it is successfully expressed. If it is not successfully expressed, then you have to redo this. So that requires luck, okay, and a little bit of time. So that changed a lot during the discovery of CRISPR-Cas9, wherein you can directly edit gene, okay? Using CRISPR-Cas9, it is cheap, it is fast, it is accurate, it doesn't rely on luck. So let me show you this GIF on how CRISPR-Cas9. So basically, they have enzyme that directly cuts. Okay, you can instruct them very accurately where to cut and where to add DNA, not necessarily relying on luck. So we will be tackling the resulting GMO here. So there's two types of GMO, although there could be more types, but we will be focusing on the two types of GMO resulting to this genetic modification or genetic engineering. First is transgenic, and this is a result of transferring genetic material between two different species. Here, for example, this is the worm, and this is the corn. You get the targeted or the desired traits gene from the worm, and then you combine it with the corn, and now you have this BT corn. On the other hand, this number 2 cisgenic, which is a result of using genetic material of same species or cross-compatible species. This is basically, it could be similar apple species, or just silencing the gene of a species or of the same organism. So it doesn't exchange or combine two different species. So again, there's transgenic and cisgenic. Now, cisgenic is different from transgenic and is safe because cisgenic plants are like traditionally bred plants. The only difference is that we are using technology compared to the selective breeding, which is really manual and takes time. So basically, the process of selective breeding is the same as the cisgenic modification. Okay, cisgenic plants are safe as traditionally bred plants. It's basically the same, okay, compared to traditional breeding. Now, let me share a quick video. Okay, let's just skip this video. Let's just go directly to our example. So are GM foods now safe? Knowing what is genetically modified food? Why do we need GM foods? How do we engineer GM food? Now, can we answer now the question, are GM foods safe with that information? So let's examine this case. So this is BT corn. So BT there means Bacillus thuringiensis. That's the scientific name of the bacteria that we targeted and used the DNA of it, basically the DNA or the gene that produces BT toxin. Okay, so this bacteria produces BT toxin, and this gene that encodes the BT toxin production is transferred, okay, to the corn. So now the corn can produce BT toxin. That's why it's called BT corn. Now you can see here the non-GMO BT corn, and since they don't have BT toxin in their kernel, they are easily eaten by the pest. Okay, especially if you have a very industrial corn farm, very dense corn that is not GMO. Imagine it's a very dense feast for the pest. But now this BT corn can produce BT toxin, which attacks the insect gut lining of this pest. Now, because they attack the insect gut lining, they die. Okay, so that's why you cannot see any pest on the BT corn. Now, the question here is it safe or not? Imagine you are eating the BT toxin here in the corn. So is it safe or not? What do you think? So a GLA, you cannot just answer it yes or no. You have to ask. Question more. Is it cisgenic or transgenic? What do you think? So this kind of GMO is transgenic because you transfer the gene from Bacillus thuringiensis to the corn. Okay, so two separate species put together, so that's transgenic, opposite of cisgenic. So why do we need to know if it is cisgenic or transgenic? Because we tend to group all GMO, all types of GMO together as one kind. That's what media portrays. Okay, even what do you call that? Organization, okay, environmental organization that is against GMO. But we have to ask: is this cisgenic or transgenic? Because if it is cisgenic, it's basically the same as selective breeding, traditional of used before by the farmer. But now it's transgenic. So now we know it's transgenic. So is there a risk about transgenic? Is this still safe? Okay, let's find out why it is safe. So it is safe because BT toxin is a protein that only binds to a specific target receptor in the gut lining, okay, of this worm. When you say gut lining in their digestive tract, okay, there's target receptor there, and it binds there and it kills them. So if the target receptor is absent, it will just pass through the gut without causing any harm. Okay, so even not all insects are poisoned or are toxic, are getting killed, okay, by this BT toxin. Now, in humans, we don't have this target receptor unless you are a worm. Hopefully not, but you don't have this target receptor. So even if you eat this BT corn with a lot of BT toxins, it will just pass through your stomach and you'll just go out after. So that's why this is safe. And this has been proven or have been tested for several decades, and this is available already in the market. This is just like chocolate, specific to a species, okay? Because target receptor are only present to a specific species. So just like chocolates, if you eat chocolates, it's okay. You're not poisoned, but if you feed it to the dog, they have receptor binds to this that are toxic, okay, to chocolates, then they die if they over consume. Now, resistant crops, aside from that, these are also another example of rainbow papaya, which is modified, engineered to resist papaya ring spot virus. So basically, almost all the papaya before are wiped out in the market because of the genetic modification or genetic engineering. We're able to make the papaya produce protein that makes them immune to this papaya ring spot virus. So is it safe or not? That's the next question. So what's your answer? You have to ask again. Is it cisgenic or transgenic? It is transgenic because we use the virus itself, okay, to immunize papaya. Okay, so that they are immune to the virus itself. So is it safe or not? That's the next question after this. It is safe. Okay, because the protein that is produced by that rainbow papaya is very specific to papaya ring spot virus. Okay, next, this is GM flavor saver or GM tomato, and it has enhanced nutrients or better traits. And this trait, they rot very slow. So is it safe or not? Look, this is 10 days, this is 20 days here, and this is 45 days. This is the control, not GMO. These are the GMO variety. You can see here after 45 days, the control, the non-GMO is already rotten compared to this. Now, it looks scary, okay? Imagine it stops rotting. It's already rotten for the control 45 days. So what happens here? Imagine it's how scared you will be if you don't know what's happening. Okay. So here is it safe or not? The question here, you cannot answer again. Is it cisgenic or transgenic? That's the next question that you have to ask. So in this kind of GMO, this is cisgenic. Okay, so this is not transgenic. This is cisgenic, meaning that they silence the genes inside the tomato itself, meaning they stop because in the tomato, they produce enzymes that makes them ripe, okay, slowly, okay, for a period of time. They produce their own enzymes that makes them ripe and then rotted at the end. Now, that DNA that produces that enzyme, or translate that enzyme, just has been silenced. So meaning that the enzyme that makes them ripens was slowed down its production. So that's why they are rotting really, really slow. They are still will rot eventually, but really, really slow. So do you think this is safe or not? This is cisgenic. This is safe, and it has been marketed before already. Now, this innate potato and Arctic apple, you can see here the browning effect. If you cut the potato, and if you cut the apple, they slowly brown immediately for some time. But for the GM potato and GM apple, they browned really, really slow. Okay, and it makes it more appetizing. Okay, and less reducing the food waste. Imagine. So, is this safe or not? This is, the question is it cisgenic or transgenic GMO? And it is cisgenic GMO. So it is basically safe because you just slow down the enzyme, you silence the gene that naturally produce the enzyme that makes them brown, react to the oxygen. So they still producing it, but very, very small amount and really, really slow. That's why they brown really, really slow here. Now, another one is this GM tomatoes that has been engineered to increase the production of the antioxidants, this purple one here. Now, is this safe or not? So the next question there is it cisgenic or transgenic? So for this one, this is transgenic. So because you allow the tomato to produce more flavanol. Okay, and this flavanol are coming from mouse ear cress here from this plant. So basically, they just produce more flavanols in enriched nutrients. So is this safe or not? It is. Basically, if you have more flavonoids, it is safe. You can easily, this plant as well, mouse ear cress, are not toxic. Okay, so if you eat this and you eat the normal tomato, it's the same thing. Now, another one is the golden rice. So this is very important, especially now because there's a lot of vitamin A deficient children. So here, the golden rice have been engineered to increase the production and accumulation of vitamin A, the beta carotene in the grains. So, is it safe or not? What do you think? They are? The question is it cisgenic or transgenic? It is transgenic. So they get the DNA or the gene from the daffodil and this bacteria to be incorporated in the rice so that they can produce beta carotene. Okay. So do you think this is safe? Based on the current findings, it is safe, okay, but it's not yet commercially available in the market. But they have trialed already in the field. Now, let's find out more about this one. Sea rise. Imagine we can, because during the climate change, sea level rises and a lot of farm are being flooded by the sea, and we have less land to farm. Now, some scientists are creating sea rice that can grow in salty water, and this salty water is will allow rice farming, okay, and create more rice. So, what do you think? Is this safe or not? The next question is it cisgenic or transgenic? And in this sea rice, this is transgenic because this crown grass that allows them to grow in sea or near seawater or salty water is transferred, okay, to the rice so that they can also be tolerant in salty water. Okay, so with this kind, so they are only able to tolerate, so the gene there, they can grow in salty water but will not affect the rice itself, okay? Because the extra salt is excreted through their either roots or stems. Now, another one, this is another development, GM salmon, and this is basically already available in the market. So what do you think this one, GM salmon, is this safe or not? Again, you have to ask, is this cisgenic or transgenic? Because again, a lot of information, they are grouping all kinds of GMO into just one GMO. There's different kinds of GMO, and we should know if it is cisgenic or transgenic. And in this case, this is transgenic. So they just basically put the DNA or the gene from Chinook salmon that that accumulates more protein or have more muscle mass, and the ocean spout gene that makes them survive in cold water, the antifreeze protein, and put it in Atlantic salmon. Now you have GM salmon that grows big and is able to survive in low temperature. So do you think this is safe or not? Okay, it is safe and available already in the market. Now, is it safe or not? So before forming any answer or deciding, always ask the relevant question on the topic first. And that ends my presentation. Thank you for listening.
The second lecture is from the College of International Relations. Okay, so now let me introduce Professor Scott. Professor Scott is an associate professor in the College of International Relations at Ritsumeikan University. He received a PhD in School of East Asian Studies at University of Sheffield. His specializations lie in media studies, visual semiotic analysis of TV news. He teaches advanced seminar, global simulation gaming, media and society, cultural awareness and communication, graduation research, media studies, and special lecture.
Thank you for attending, and I will try to make this interesting. So this is a short session which I've prepared for today. It's actually a part of a class that I do on a course called Cultural Awareness and Communication, which is an undergraduate course for second and third years in the College of IR. So it's available to JD students and GS students as well, who study alongside them. If you want to look at the slides online, there's a URL here, and also the QR code should take you to the slides on Google Slides. So if anybody wants to grab the slides and look at them independently or download them, then please do so. And I can put this QR code up again at the end if anybody's interested. Okay, so I'll give you a few more seconds to grab that. Okay, so yes, the subject today is stereotypes, prejudice, and identity. And these are quite big questions, quite big topics. So I'll start off by saying that we're not going to talk about all types of identity. As far as academics are concerned, there are different types of identity. And it's helpful if we can start by narrowing the nature of this thing, identity, down to something more manageable and certainly more actually connected to the type of study that we do in this cultural awareness class. So we're not interested really in kind of literary identity or psychoanalytical identity. We're interested in not so much either what we call psychological identity, because different areas of academic work have different uses of the word identity. So the one we are interested in this class and in this particular session is what's called social identity. And this is the kind of identity that emerges from thinking about what kind of groups within society we belong to. And it's often our membership of these different groups, whether we think we belong to them or whether other people put us into them, that create this thing called social identity. So it's this idea of social identity that I'd like to talk about primarily today. So if you know things about the other types, psychological identity or psychoanalytical identity or the other things, forget those. We are concentrating on social identity because this is a class about culture. And culture is ultimately a social thing. So we're interested in how identity works in a social context. Okay, so next slide. You might be wondering what kind of relationship there is between the idea of stereotyping and the idea of social identity. But I hope by the end of the session, you'll see that there is quite an important relationship, which it helps us, if we understand it, to understand kind of how our societies can work in certain situations. So stereotyping, ultimately, stereotyping it's the process of ascribing characteristics to people on the basis of their, I put apparent, group memberships, apparent in brackets there, because we're often relying on how people appear to be to decide which groups we think they belong to often. So there are some very, very typical kind of stereotypes. These are not my opinions. These are just kind of general sort of social stereotypes, which you may have come across. You know, for instance, things like women are emotional, or men are aggressive, or Italians are romantic, or Japanese high school students are hardworking. All these kind of ideas that we have about certain groups within society. You could also say, I don't know, university professors are kind of eccentric. Who knows? There are all kinds of fixed ideas that we attach certain attributes to certain groups within society. And we do this often on the basis of identifying and categorizing people into different groups. So stereotyping is not a kind of fixed thing. It's not something where there are fixed ideas in society about how particular groups are. It's a process. It's something that you do. Stereotyping is not something that is, it's something that people do and actively engage in, okay? And this is my attempt, using a very exciting diagram, to explain roughly what that process is. I mean, I'm sure if we were to look at this in terms of brain function and in terms of a more kind of psychological, scientific approach, it's probably far more complicated than this. But for the kind of social level of thinking that we're doing today, let's say that stereotyping is a process where we identify people who belong to or seem to belong to a certain group. You say, ah, that person over there, they seem to belong to this group that I think exists within the world, whether it's a gender group or an ethnic group or some kind of social group or who knows. But you say, I think that person belongs to that group from their appearance, from what I can see of them. And then outside knowledge and things we have learned through our kind of process of growing up within a society come into play. And the next step is to think, aha, I know about people who are in that group. And people who are in that group are like this. They have certain attributes, they behave in certain ways, they think certain things, they believe certain things, okay? So we have some knowledge of, or some guesses about a particular individual. We have some knowledge or guesses about a particular group who we think they belong to. And the final step to stereotyping as a process is this putting these things together and reaching the conclusion, it might be right, it might be wrong, but we reach a conclusion that if this person belongs to this group that I know about, then this person is like this because they seem to be a member of this group. Okay, and that's a kind of simplification of the process of stereotyping. Identifying individuals that belong to a group, thinking we know something about that group, and then transferring that knowledge onto the individual and saying, this person is probably like this because they belong to this particular group. Okay, I hope that makes sense. So it's stereotyping is not a thing, like I said, that exists. It's a thing that we do all the time. So it's a process. It's something that happens. There are lots of different sources of knowledge about groups within society. And within any particular society, there are a whole loads of ideas about how particular groups are, okay? And these are referred to as social stereotypes. These are not stereotypes, kind of ideas that we have kind of created in our own minds through our own experience. These are things which we learn from the society around us, whether it's through education or through, talking to our peers or through engaging with the media, for instance. We develop a set of ideas about how particular groups are, and these are often shared amongst other people within our society. So our society ends up with a set of ideas about how certain groups are, okay? And this general phenomenon we call to the sort of creation and recreation of things called social stereotypes. This is how our society creates images of different groups. And there is a tendency, a very normal human tendency, to assume that people within the same group are similar to each other, and people in other groups are different. Okay, so the people within group, our group, for instance, are more similar to each other, and people who are not in our group are more different from us. Okay, and I explain how we kind of understand this when I come to the second part of the class, which is about an experiment. There's also a tendency to assume that groups that we belong to, or groups that I belong to, in this case, sort of abstract I, me, groups that I belong to are somehow better than groups that I don't belong to. Okay. And these two fairly simple ideas, the idea that my group is better than other groups, and people in my group are more similar to each other and very different from people in other groups, it's not something that is kind of the figment of an academic imagination. It's actually the result of experiments and studies done in various fields, including kind of psychology, experimental psychology, to actually investigate how people see the world and how they kind of arrange ideas about the world and how they understand social relations. But I'm kind of contracting a lot of this explanation because this is quite a short session. So I apologize if it's a bit, you just have to kind of trust me on this one. Okay, so stereotypes are these I kind of group ideas about how different groups within a society, the sort of attributes and their behaviors that different groups have. Okay, if we look back to early studies of stereotypes, so done about 100 years ago, and written about by people like this chap, this is Walter Lippmann, who was a very well-known American kind of journalist, philosopher, public intellectual, and he wrote a book called Public Opinion, which I'll give you a link to that because it's on the internet and you can download it and read it if you're interested. He wrote a book called Public Opinion, and where he started to think about in a fairly organized, systematic way about what stereotypes are, their relationship to things like public opinion and journalism. And he described the knowledge contained in stereotypes, so these ideas about groups shared amongst our society, in these kinds of terms. So he said the knowledge is partial and inadequate. So the knowledge of other groups that we have in societies tends not to be very deep. It tends to be just segmentary. We know little bits about other groups. And it's generally what he called inadequate. It's insufficient for us to truly understand what another group is like. Some of the things he said are quite controversial, maybe, and modern psychologists, modern people in cultural studies may not agree with his words entirely, but it's a good starting point for thinking about how understanding of stereotypes has developed over the last 100 years. He also said that stereotypes or knowledge kind of based in stereotypes was defensive and precluded objective, balanced reasoning. So he said that when people have ideas about certain groups, it's very difficult to change their minds by reasoning. The knowledge in stereotypes and knowledge of other groups is often deeply emotional in some ways, and therefore reasoning and factual arguments tend not to change people's minds about what they believe about other groups. So in that sense, he suggested that the knowledge contained in stereotypes is what he called obdurate. So it's very difficult to change. And one of the reasons he was interested in stereotypes and described stereotypical knowledge in this way is because he was interested in the link between discrimination, in particular, so discrimination against women, discrimination against different ethnic backgrounds, particularly Black people in America in the 1920s and 30s. And he linked the idea of discrimination or prejudice within our societies to stereotypical thinking. So people who think in a stereotyped way, who do this process of stereotyping other people, are often also prejudiced. And he said there's a kind of link between prejudice and stereotyping. Okay, so he's interested in this link. And so lots of the early studies of stereotyping were attempts to identify the kind of content, what we call the content of stereotypes. So if you see an individual who appears to belong to a certain group, what kind of ideas do you have about that person when you see them? And a lot of early studies were trying to identify the content. So what kind of ideas did certain groups have about certain other groups within society? And the link between stereotype thinking or stereotypical thinking and prejudice kind of created a sort of moral story about stereotyping, in that it was thought that people who engaged in stereotypical thinking were prejudiced and therefore bad. and there was a certain sort of division between the good people who don't stereotype and bad people who do stereotype. Okay, and so the idea of stereotyping and this link between kind of prejudice and social problems became a focus for lots of study on stereotypes in the early days. And this link didn't really go away. It's not now what we think, but this link didn't really go away until the 1960s, when people like Henri Teifel and his collaborators began to look in a more scientific way, in a more using kind of experimental psychology, to investigate how the actual mental process of stereotyping worked. He came to the conclusion that in this book that I've mentioned on the slide, Cognitive Aspects of Prejudice, that human minds work by categorizing the world, simplifying things into categories. And this is a normal function that all human minds do. So there's not a link between necessarily classifying the world and being prejudiced. It's just something that all human beings do, because that's the way the human mind works. And it might, well, you might ask fairly reasonably, well, how on earth did he know? And how on earth did he manage to kind of argue that this was true and sort of prove it, if you like? He came up with an extremely simple and effective way to investigate human reasoning about categories, about putting people into groups, if you like, about putting things into groups. He's not the only. This is not the only experiment that was done. There are lots of others which have, replicated these kinds of results. But it was his contribution to our understanding was simplifying the idea of prejudice down to a very, very kind of simple process that could be investigated using a scientific methodology, a repeatable, reproducible methodology. So a way of doing the experiment that anybody could test and, get similar results. Probably what I'm going to show you now will surprise you, because he didn't use human beings. Human beings are very, very complicated things. So what he did was he tried to find almost the simplest things there are and see how the human brain categorizes things. And what he used was some lines. These lines. These are very special lines, which I borrowed for this lecture today. I have to return them to the original owner later. But these are the lines that he used, he and his collaborators used. And these are special lines because they increase at a regular rate. The shortest line, 16.2 centimeters, and the longest line, 22.8 centimeters. And in between, there are a number of lines, and they all increase by 5% in length. Okay. So you're thinking, what on earth can a bunch of lines, how does that help us investigate stereotyping and social prejudice? Well, this is the experiment that he carried out. And you see, it's a very, very clever experiment and a very clever way to simplify this mental process and investigate this mental process of putting things into different groups. So using these lines, what he did was he trained a number of observers, a number of people who would, look at the lines during the experiment, and he trained them to guess the length of the lines. Okay, so this is quite a hard thing to do. So over several weeks, he trained a number of observers to estimate the length of the lines, and that's all they had to do. And so when it came to doing the experiment, he had what we call an unclassified case, unclassified condition. And during this portion of the experiment, there's an extra portion that comes next. During this portion of the experiment, what he did was he showed lines on a piece of cardboard to individual observers who had been trained and asked them to estimate the length of the line. So that's very simple. Yeah. It's a line. People are trained to estimate the length of the lines, and he asked them to do what they've been trained to do. Okay, so in this first portion of the experiment, the unclassified condition, he got a set of estimates from a number of observers for how long these lines were. Then, this is the clever part, he did the same experiment again, but labeled the longer lines B and the shorter lines A. Okay, so the lines were exactly the same lines, and it was the same observers. However, in this, what he called the classified condition, the lines were labeled A or B. Okay, nothing else changed. And obviously the intention here is to see what happens when you put things into groups. In the first condition, the unclassified condition, the lines are not in groups. They're just lines. They don't seem to belong to, one group or another. They're just lines. In the classified condition, the lines are in groups. There's a group B, which is longer lines, and a group A, which is shorter lines. And that's the only difference. And what Teifel and his collaborators discovered when they looked at the results was this. Okay, so I'll just give you a moment to actually look at this. I'll explain what the table means. Okay, so in the first column you have the group which the lines were in. In the second column there's the actual length of each line. In the third column there's the difference from the next line in I believe millimeters centimeters. In the third column is the difference between the lines, which was calculated from observers' estimates of the lines. Okay, so you've got column 1 is the real length, column 2 is the difference, column 3 is the estimated difference, which is calculated from people's observations. And the last column, the column on the right, is the relationship between the real difference and the estimated difference. Okay. Okay, so column A, classification, and then the other columns as labeled. And the results, well, if putting lines into group A and group B didn't make any difference, then it would be very difficult to explain the number that I've highlighted in red. I hope you can see that. It's, let me see, 1, 2, 3, 5th row down, including the header, at the number 2.1, which means that the estimated difference between the longest line in group A and the shortest line in group B was double what it actually was in real life. Okay, so if we measured the longest line in group A, which is the short lines, and the shortest line in group B, which is the longer lines, then when people estimated the difference between group A lines and group B lines, the kind of border between the two groups, they estimated the difference to be twice as much as it actually is. And if you compare that with the other numbers in that last column, so 1.4, 0.9, 1.3, they're all about I'm going to average about 1, which means that the estimation of the difference between the lines is kind of fairly accurate and almost, close to the actual real difference. If all the numbers were 1, that would mean that everybody had estimated the difference between the lines perfectly. Obviously, that didn't happen, and there's some fluctuation around 1. Some of them are shorter, some of them are longer. But the number between the shortest B line and the longest A line is by far the biggest, 2.1. The next biggest is the end of the B's section, so the longest B line, if you like. So the gap, the kind of borderline between group A, short lines, and group B, long lines, is the place where the difference in people's minds is exaggerated the most. And what Teifel concluded from this, and not just from this, but doing other kind of experiments as well, was that if you label things in the similar way, then the human mind perceives them as being more similar to each other. So if you say that some things are in a group by labeling them somehow, then the human mind automatically kind of assumes similarity and perceives them as more similar. And if you label things differently, then the human mind automatically assumes that they are more different than they actually are. To put this in sort of scientific terms that Typhon and his friends uses, intragroup similarities, that's the similarities between things in the same group, look more similar. If we think things in a group, then our mind kind of tends to perceive them as being more similar to each other. Whereas if we think things are in different groups, then the difference between the group we know and the other groups tends to be also emphasized. So it tends to be exaggerated in our minds. So if we put that in kind of human terms, what this tends to mean is that if we perceive ourselves or think of ourselves as being in a particular group, we are more likely to think and perceive other members of that group as being more similar to us or more similar to each other, including us, than they actually are. Okay. So if we think everything, say we're a member of group A, then we will perceive other members of group A as being more similar to each other and more similar to us, because we know, our mind knows, that they are in the same group. On the other hand, if we encounter people who are maybe in group B or group C, then our perception, because of our knowledge that they are somehow in a different group, means that they will be perceived as being more different than they actually are. So we feel closer and more similar to the people that we think we're in groups with. And this kind of phenomena has been used as the basis of lots of different experiments, including some in sort of colleges and schools where, for instance, children are put into groups and then asked to carry out some performance. So there's a very well-known example where I don't know if you've ever done peer marking in schools, where you as a pupil mark other pupils' work. There's a very well-known example where students were put into two groups, group A and group B. They were chosen at random. There was no difference between them, and asked to mark each other's work. And you can guess what the results were. People in group A gave better marks to other people in group A and gave worse marks to people in group B. Obviously, there's a bit of kind of statistical manipulation in there to make it all kind of make sense, but there is a tendency to favor people who we think we're in the same group as. And again, this has been sort of illustrated again and again and again and again. So ultimately, what I want to say is, having taken you on this kind of excursion into the human mind, is that we as human beings kind of have to be aware of these things, because our identity, who we think we are and who we think other people are, is not just a kind of matter of looking at what people are and do and sort of putting them into simple groups. Sometimes the group that you belong to isn't the group that other people think you belong to. You can make certain choices about communicating group identities, but often you don't really know what other people think your group is like. And so some of the things that happen to us in society that may be inexplicable, we can only really understand by knowing about our group, knowing about other groups, and knowing about these kind of ideas that exist in kind of these sort of shared social stereotypes and shared ideas that other groups have about us or about themselves. And so this is not necessarily evil or bad or immoral, or it's not something that we can prohibit, because it's something that our minds do fairly naturally. However, now that we know that, we can kind of move on to the next step. Now that we know that our minds are doing this, we can begin to kind of be more self-aware, and that's one of the reasons why this class exists. It's called cultural awareness and communication, and one of the reasons this class exists is to give people give students tools to become aware of these kind of things that go on in the world, to become aware of their own kind of personalities, their own identities, and the way they relate to other people, and also to understand themselves as human beings, because human beings aren't merely kind of social animals. We have shared physiology and shared kind of bodies and minds that match up with other people around the world. So understanding how human beings work is also an important part, because, like I hope I've demonstrated, perception of other groups is not merely a social thing. It's also a kind of a cognitive, mental thing that human minds just do. But hopefully learning about these things gives students tools to think about how they relate to other people and how they relate to themselves, and to be aware of the pitfalls and problems that not being careful about how we think about other groups can lead to within our societies. So ultimately, it's about awareness. As I said, the course is cultural awareness and communication. So it's about awareness of things like how human brains work, how human groups work, how human societies tend to operate. And if we can do that, then hopefully we can take a step away from being thought of as prejudiced or acting in a prejudiced way, and we can start to treat people with fairness and respect that we would want for ourselves. So ultimately, that's my aim, is to give students tools to think about who they are, what their society is like, and also to act in a way that makes those societies hopefully better, more equitable, more amicable, and kind of more fun for everybody. So I hope that's the end of the content. I hope it was interesting for everybody. I apologize for having kind of abbreviated a few things, but I hope it was interesting and gave you something to think about. So thank you for listening. And if you are interested in doing any further reading, as I said, you can download most of these things. Most of them are available online, so you can download most of these things if you want to find them. There's some interesting books. Teufel's work is, I think, maybe online, and certainly Walter Lippmann is. That's a bit old now, but a good place to start. And if you want the QR code again, there it is. I hope that was interesting. Thank you very much.
So the last lecture today is from the College of Policy Science. So Professor Miya is an associate professor in the College of Policy Science at Ritsumeikan University. She received a PhD in architectural engineering from Osaka University in 2014. Her research focuses on urban planning and community development through architecture. Her specializations are urban planning and community design. And she currently teaches advanced policy studies and survey analysis.
So my name is Miya Yamade. I am member of CRPS measure. And I'd like to do demonstration class for around 30 minutes. So today's lecture title is Why Community Design Now? So that means why community design is important now. So this is to consider post-disaster community reconstruction and case of Fukushima after radioactive contamination. So in this course, in this lecture, students learn the current situation as well as issues about urban cities and measures to mobilize students to build a sustainable society. So learn the current situation is Fukushima and measures to mobilize students to build a sustainable society means community design methods. So the three parts will be explained today. So one big question for you. So do you want to eat vegetables from Fukushima? So have you ever considered these questions? So the end of this course, I will ask same question. So I hope your thinking and your idea will be changed. had changed. So we have to know, so what happened in Fukushima? So in March 2011, we had large earthquake in Fukushima. Name is East Japan earthquake. So after that, nuclear accident happened. So communities were divided by radioactive contamination from this time. So as we can see from the figure 1, so pink color shows operational plants and green color shows shut down plants in the world. So what I want to say, so the same situation as Fukushima could happen at any time in the world. And so this is current situation and issues. So did you see this newspaper article from last August? So treated water from Fukushima plants being released into the sea. So we have still stigma about food products, including seafood. So what is stigma? So the stigma is the phenomenon of people not buying food or drinking water due to negative perceptions. But it is true or not, we have to think about ourselves. So what types of stigma exist? So for example, not buying food and eating food from Fukushima, or indifferences means I don't care phenomenon. And established negative image. So this problem, this issue, including they don't buy food from Fukushima. So it is true. So we have to think about why does stigma exist? For example, lack of knowledge or negative image or miscommunication. So we need to identify the facts by ourselves. So it is important to study community design method. So if you join our class, our seminar, so you can learn how to protect, how to decrease, how to so how to disappear stigma using community design method. So I say many times why community design now or community design is important. So what is community design? So I have to explain. So creating how people connect with each other and how they work together. So why community design is important now? So maintain and develop communities. So independence, so sustainable society. It not rely on government support. So we can do ourselves. So many people living together, so help each other. So next, why the interview survey is important. So from here, I will explain what my seminar, what we do. So we focus on farmers' guest houses in Towa District, Nihonmatsu City in Fukushima Prefecture. So where is Towa town? So and where is Fukushima? Maybe you know Fukushima, but you don't know small town of Towa. So I will explain. So this is Tokyo here. So our school is here, Osaka. So Fukushima is around here, near Tokyo. And Fukushima nuclear power plant is here. And that color shows contamination area. So my research focus on this area. Name is Towa Town. So what I want to say, so this is not into contaminated area, but they have so severely stigma image, so bad image. So if they made food, they many people doesn't buy that. So what happened in Towa area? So here is farmers' guest houses around 225 owners. So I will show you, I like this. So 25 owners here. So I will show you a short movie now. I'm gonna love you till the end of time. Yes, so there are 25 owners keeping their house for guests from other areas. So they want to learn what happened in Towa Town in Fukushima. So they want to know the current situation and how to protect, how to continue their community. Also, I went to there many times for 10 years and asked the people, do you need help? Do you need, so through interview survey. So I will explain about key questions on the interview survey. So how can we decrease stigma against agricultural products in Towa Town, including Fukushima? So how can we use farmers' guest houses effectively? So it is important to understand residents' opinions. Yes, this is the interview survey I will show you. So let's conduct an actual interview survey to understand the current situation and propose a community design method that match the region. So if you join my seminar, so let's explore a better community design using interview survey. So also I will plan to interview survey is for this woman. So she is an agriculture and we know that to understand the current situation and to identify current issues in the community and to decide objectives of the survey and consideration of question items. So in this lecture, the most important thing is to listen to and understand resident opinions. And a little bit I will explain this lecture what we have to do or what we will do and the skill you can gain in this class. So I will show you some course schedule here. So lecture 1 to 3, your study current situation in Fukushima, and also discussion time we have. And the skills you can gain in this class is to identify local issues. And lecture 4 to 7, so learn the methodology for community design. So later I will explain about big data analysis, but here, so skills you can gain in visualize opinions is to explain others. And also we have an interview survey. We go to this area and using online or something, and then you can gain ability to organize residents' ideas. And finally, we have final presentation and group work and discussion, and also skills you can gain to combine ideas to others like this. So we have 4 parts in this lecture. So I will explain a little bit. So how to visualize consciousness of using text data analysis. So we can know so public perception using SNS data or text data from newspaper article and thesis or something to identify the differences between public perception and information source. So then we will compare the contents between SNS and newspaper. So we know public perception, the people, what do think about Fukushima contamination, about situation. So when we use text mining, it is the soft name KH coder. So we can see the text for visualized like this. So we have to compare, we can compare the SNS data and the newspaper article, what they think about, what is different or same ideas. So this is visualize the text data. I hope you enjoy my presentation. I hope so. So thank you for your time. So, and if interested in my research, so you can join Instagram here.
Okay, so thank you again for joining our mock lectures today. We hope the lectures were interesting and help you get a better idea of what it's like to study at Ritsumeikan University. Here is our contact information. If you have any questions, feel free to reach out anytime. You can also book an individual online meeting and ask questions directly to the admission officers. Information on future events is available on our website, so we hope to see you in one of them or maybe at Ritsumeikan University. Thank you very much.